Welcome to this fascinating journey into evolutionary biology. Today, we are going to explore the molecular evidences of human evolution. Have you ever wondered how we know exactly when humans and chimpanzees diverged, even without a complete fossil record? The answer lies hidden inside every single cell of your body. Let us dive in and learn how to read our genetic history. To begin, imagine you find two nearly identical instruction manuals for building a car. If both manuals have the exact same typo on page 10, you would immediately know one was copied from the other, or both came from a master copy. In evolutionary biology, our DNA is that instruction manual, and genetic mutations are the typos. Because these mutations accumulate naturally over time, comparing them gives us incredible insights. When we look at human and chimpanzee DNA, they are nearly 99% identical, sharing the exact same typos in the exact same places. This is not a coincidence. It is a direct, readable record of our shared ancestry. Now that we understand the manual analogy, let us look at the scientific field that studies this, molecular phylogenetics. Instead of just looking at the physical shapes of fossilized bones, scientists sequence the exact order of nucleotides in our DNA, or the amino acids in vital proteins like cytochrome C and hemoglobin. By comparing these sequences across different species, we can build a highly accurate family tree. This powerful toolkit has definitively proven that chimpanzees and bonobos are our closest living relatives. Furthermore, by extracting ancient DNA from preserved fossils, scientists have even mapped our intimate relationships with extinct cousins like the Neanderthals and Denisovans. You might be wondering, why do we even need molecular evidence if we already have a fossil record? The truth is that the fossil record is incredibly fragmented. Bones only fossilize under very rare and specific environmental conditions, which leaves massive gaps in our historical timeline. Molecules, on the other hand, are present in every single living creature today. They provide a continuous and highly detailed map of our evolutionary journey. Without molecular biology, scientists would still be arguing over our relationships based purely on physical traits, which can often be misleading due to convergent evolution. DNA gives us an objective, mathematical truth. This brings us to one of the most brilliant concepts in evolutionary biology: the molecular clock. To figure out exactly when humans and chimpanzees split from their common ancestor, scientists rely on neutral genetic mutations. These are changes in the DNA that do not affect the organism's survival, meaning they are not filtered out by natural selection. Because of this, they accumulate at a constant, predictable rate over millions of years. Think of it like the steady ticking of a clock, where every tick is a new mutation permanently recorded in the DNA sequence. By counting these ticks, we can peer back in time with incredible precision. Now that we understand the concept of the molecular clock, let us look at its mathematical form. The elegant equation we use is D equals 2 times R times T. Let us break down what each symbol means. D represents the total genetic distance, which is the number of base pair differences we observe between two species today. The symbol R represents the mutation rate, meaning how many mutations occur per lineage every million years. Finally, T represents the time since the two species diverged from their last common ancestor. If the mutation rate R increases, the species will accumulate differences much faster, leading to a higher genetic distance D in the same amount of time. Before we move on, let us see this formula in action with a concrete example. Fossil evidence suggests modern humans and Neanderthals diverged about 0.5 million years ago. Let us assume the mutation rate for a specific mitochondrial gene is 12 mutations per million years per lineage. We want to predict how many genetic differences we should find. We start with our formula, D equals 2RT. We plug in 12 for the mutation rate R and 0.5 for the time T. Multiplying 2 by 12 gives 24, and half of 24 is 12. This means if we sequence this gene, we expect to find exactly 12 base pair differences, which perfectly matches real-world ancient DNA tests. So we have successfully used the formula to predict genetic distance. But what if we want to find the time of divergence? Let us understand how we rearrange this equation and why that factor of 2 is so important. We start with our base equation, D equals 2RT. The reason we multiply by 2 is that after the evolutionary split, both lineages are accumulating mutations independently at the exact same time. The human lineage accumulates R times T mutations, and the chimpanzee lineage also accumulates R times T mutations. Adding them together gives us the total genetic distance D. This setup perfectly balances the total change's genetics against the rate of change. Now that we understand the setup, let us isolate the time variable t, since time is the unknown historical factor we usually want to discover. To do this, we divide both sides of our equation by the combined mutation rate of both lineages, which is 2r. The 2r cancels out on the right side, leaving t completely by itself. This gives us our final rearranged formula, t equals d divided by 2r. This algebraic rearrangement is crucial because it beautifully transforms our raw genetic data, the distance d, into a readable historical timeline. We are now ready to use this to date human evolution. Let us apply our newly derived time formula to a real scientific question. Suppose scientists find a total of 30 mutations between humans and chimpanzees in a specific DNA region. The mutation rate is 2.5 mutations per million years. We want to calculate when we shared a common ancestor. We use our formula, T equals D divided by 2R. We substitute 30 for D and 2.5 for R. The denominator becomes 2 times 2.5, which is 5. Dividing 30 by 5 gives us 6. Because our rate was in millions of years, our answer is 6 million years ago. This mathematical result perfectly aligns with physical fossil discoveries. Now that we have calculated these divergence times, let us visualize them on a phylogenetic tree. Look at the vertical axis on the left, which represents time in millions of years ago, counting down to the present day at the top. Notice the branch for gorillas splitting off first, around 8 million years ago. Moving up the trunk, we hit a critical node at 6 million years ago, marked with a clock icon. This is the exact divergence point we just calculated between humans and the chimpanzee family. Further up, at 2 million years ago, you can see the bonobos and chimpanzees splitting from each other. This diagram perfectly illustrates how the molecular clock maps our evolutionary history. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that our DNA acts as a living history book, where genetic mutations serve as a readable record of our ancestry. Second, we explored the molecular clock, which relies on the constant accumulation of neutral mutations to precisely date evolutionary splits. We use the formula D equals 2RT to mathematically link genetic distance with time, proving that humans and chimpanzees diverged about 6 million years ago. Finally, we saw how this objective, quantifiable molecular data perfectly complements the fragmented fossil record, giving us an undeniable picture of the human family tree. Great job following along.